the basic system utilize CRISPR-Cas or Cas elements of the system, and then guide RNA Cas9 usually linked to a transcriptional activator, and in our case, to transcriptional repressor to downregulate gene of interest, in this case, APOE. And the new development, which we would love to report at this conference, is that we develop system to fit all-in-one epigenome-based components of a CRISPR-Cas into adeno-associated vector. The study evolved from, as I mentioned, a innovative approach of epigenome editing, downregulation of APOE, and we evolved system to fit again to a adeno-associated vector as a platform of preference for us through a comprehensive screening of Cas9 and repressor combination. So the idea was the main challenge here, that adeno-associated vector is very small vector, and CRISPR-Cas is very bulky system. So we had to screen to find best combination between Cas9 and a repressor, which will fit a size of AAV without compromising on the titer, potentially which fit a clinical study as we plan in the future based on this system. So this combination was found through a comprehensive screening, as I point out, and we were able to successfully create a all-in-one AAV based on Cas9, small compact Cas9, paired with a repressor, and we use a synthetic repressor to be linked with Cas9. And the study focused first on validation of this system in vivo, as you correctly pointed out. And to that end, we simply validated this system on ability to repress first unrelated reporter of GFP, in our case, driven by CMV promoters. So the idea was, let's first get proof of concept and validate the system in vivo on unrelated to APOE gene, on something that would be really feasible and easy to detect as a green fluorescent protein. And we found very efficient downregulation using this system, meaning the GFP expression was downregulated by close to 100%, 90-95%. Very impressive results, which support our idea, and we move the system to more interesting discovery, more interesting proof of concept experimentation, in which we specifically targeted APOE promoter region in mice. And fortunately here at Duke, we have mice, so-called humanized mice, which are knocked in with human APOE. So they don't have a mouse APOE, they have a human APOE, and specifically APOE3 or APOE4. And using these mice, applying our system, we were able to demonstrate great downregulation of APOE gene. Very impressive, close to 70%. We also, and again, it's something that I'm going to present as well, validate this system on human organoids, which derive from patients with APOE4 or APOE and APOE3 genotype. So the system definitely worked very efficient, and we're really happy to report this on this meeting. as much as we can get proof of concept results for this study as beneficial for us, as the ultimate goal is to move this program to clinical research, to clinical trial. We created a IP based on this study, and at this point we trying to attract funds to move it all the way to clinical research. But you're absolutely right on the point that first, as we will go to FDA approval and pre-IND studies, we definitely need to validate this concept, this novel concept, and those novel results in a large animal models such as non-human primates, which we are playing. So in parallel, I would like to mention that it's not easy, as there's no good animal model in human primates exists for to apply and to test any drug development. There's no good AD animal model in primates. So on this end, we also, and I'd like to share with you, so our research focused on creating such a model based on viral vectors. So what we did and what we are doing at this point, we trying to create this animal model by integrating into viral backbone. And again, we will use AAV for high efficiency, so-called Swedish mutation in APP, and it will be combined with non-tau mutation in tau gene. So we're planning to use this system to inject it as to induce a AD-related phenotype, and then apply our therapeutic system to try to rescue this phenotype in non-human primates. So this is next step.